The purpose of this video is to go over how to approach identifying an unknown bacteria from a list of known bacteria in a typical microbiology lab. The typical approach is to have this as one of the final labs of the year, and the unknown bacteria will typically be one of the bacteria that's been tested in the lab over the course of the semester. So how do you approach designing a protocol for identifying this unknown bacteria? That's what we're going to discuss. Keep in mind that as you develop this protocol, you need to do as few of the microbiology tests as possible in order to identify the bacteria. Some professors will even put a limit on the test. So we will review over how to pick the correct test and which tests to avoid and which tests are good solid tests to run. As silly as it sounds, the reasoning and the thinking process that goes into approaching solving that unknown bacteria is very similar to the reasoning process that goes into playing this Guess Who game. So let's look at this Guess Who game for a second. Just kind of bear with me and follow along, and I'll tie it all together. But as we look at this list of individuals, if we wanted to take this large group and divide it down into two groups that would be workable, what would be a way we could break this group in half? Keeping in mind that one of these individuals is our unknown. Well, what I would do is divide these into two very obvious groups, women and men. And so I've listed each of their names, and I put the women under one category and the men under the other. And so now let's just look at the women. We have this unknown individual. We've determined that it's a woman. So how are we going to identify specifically who it is? Well, we will no longer need to look at the men, as our test, male or female, ruled out all of those males. And so all of the individuals that are listed as males will be discarded from our list of possible unknowns. And so our unknown is one of these right here. So we now need to take this group and divide it down into smaller groups, preferably where we can divide it in half. So we'll say, okay, of this list of women, do they have a hat? Well, yes, 2 of them do, and 3 of them do not. Again, that divides that list in half. The women that do have a hat, we could look at it and do a third test. What is the color of the hat? If it's red, we've identified it as Rachel, and if it's green, we've identified it as Ashley. So we have identified our unknown person in as few as 1, 2, 3 tests if it's one of these two. Now, if we determine that the individual did not have a hat, we'll discard these 2 people and only look at these 3. Okay, so let's look at something that we can divide these 3. How about glasses? Okay, if they have glasses, then we've identified as Emily, and if no, we've narrowed it down to 2 possible choices. Okay, between these 2, we can go with hair color. If it's blonde, we've identified as Megan, and if it's black, it's Sarah. So if our unknown was one of these 2, we identified them in as few as 4 tests. And so if we had to identify our unknown and they were a male, we can look at the next page. I'm not going to spend as much time on that, but I did the same approach for the males that I did as females. There are more males, so you will notice that when we get to the next page that it is a little bit longer, but each time I take the group and I try to divide it in half by doing a single test. And so we had a much larger group of males, so we would need to do more tests to identify. And we could look at, does the person have facial hair? We divided them into half. Yes, they do, and no, they don't. If the person does have facial hair, all of these individuals are discarded. We'll then take these individuals and then see how we can divide them in half. Is that facial hair a beard? If yes, we've identified the individual right here, and we can discard these individuals. Okay, now we need to determine this individual. Do they have hair? Okay, if yes, we've identified it as hair, and we can discard these. Okay, of the two that have hair, is the hair curly? If yes, then we've identified the individual, and we identified him in as few as 5 tests. If you have taken a college or a high school biology lab that's heavy on laboratory experiments, you have done this for many years, and this is called a Dichomous Key. So basically what you're doing in microbiology to identify your unknown is you're developing your own Dichomous Key that you will be following. And here's just a picture, and you could practice making one for this, but let's see how we would approach our unknown bacteria. So you're going to use that same reasoning as you approach microbiology. And the first test I would do is always do a gram stain. The gram stain will take your list of bacteria and divide them into either gram positive or gram negative. Just like we started with our grouping of people, we divided them into either male or female. And what this does is this then tells us, okay, this unknown bacteria is gram positive, and I can ignore all of the gram negative bacteria. Or this unknown bacteria is gram negative, let's ignore all of the gram positive. And then once we've done that, we'll take that list and then we'll look at another testing we can do that will group the bacteria into a smaller group. So let's look at some examples. And as we look at the examples, just continue keying out your organism until you have identified that unknown. Now here is the list of bacteria I typically use in my microbiology class, along with the various test agents I teach my students. And my bacteria actually takes 2 pages to list. So this is the page 1 of the bacteria we work with. Here we have page 2. It's the same test, just 2 of the pages so I could continue this down. We're going to first list the possible bacteria. So here I have the possible bacteria, and I believe there's 9 there that we work with in the course of my micro class. And so the first thing I would do is I would take this 9 bacteria and I would set up my Dichomous Key. So what I would do is say, okay, let's divide these up into gram negative and gram positive bacteria. And here I have that 3 of these are gram positive, and I've got the remaining ones are gram negative. So my unknown bacteria, I run the test. If it's gram positive, I have now discounted all of these bacteria, and I'm now just looking at 3. Now let's say that our unknown was a gram positive bacteria. Okay, so it's one of these 3. Now I'm going to go back and I'm going to look at the possible tests that I could run, and I'm going to see what could I do that would break this down. And upon reviewing over my sheet with all of the possible bacteria and their test results, I see glucose fermentation. It's a wonderful test to run. And if I have a positive glucose fermentation test, it's either Bacillus subtilis or Staphylococcus epidermis. If it's negative, I've already identified my unknown as my Micrococcus lutea. All right, let's say that my unknown bacteria was positive for glucose fermentation. I'll then look and see, okay, what test could I do that would differentiate these 2 right here? And I go back and look at my sheet of the tests that we've ran and the test results, and I see that lactose fermentation, each one had a different result. The Staphylococcus epidermis was positive, and the Bacillus subtilis was negative. So I come up with my key, I write this out in advance, and I would go back and I would write one out for my gram negative bacteria, separating it down just like I did with the gram positive, so that I then go into the lab. I run my first test. It's a gram test. I look at my results. If it's gram positive, then I'm going to follow this key through. If it's gram negative, I'm going to ignore this stuff up here, and I'm going to follow the key that I made here, so that a gram positive, I know, okay, my next test I want to run should be a glucose fermentation. Okay, my glucose fermentation was positive. Next test I need to run, lactose fermentation. If it's positive, I've got my Staph epi. If it's negative, I've got my Bacillus subtilis. So I've worked out in advance what test I should run based on the results of the previous test. And so I just go in and follow my key until I key out and identify my unknown bacteria. Now, do keep in mind that some microbiology lab tests are better than others, and I do have 2 videos on the microbiology lab test. You can see them on the YouTube at Kim Owen or on my website, kowenscience.com, and you can see which tests work best. But these are the tests that I typically run, and you'll notice that gram stain is number 1 because that's the most important one. That's what I would always start with for identifying the unknown. Motility is not a very reliable test, and if you can key out a bacteria by not using motility, I would do so. Also, notice right here EMB and McConaughey. Each of these tests for a gram-negative bacteria, and they each test for whether the bacteria ferments lactose. These do the exact same test. So I would not run an EMB and a McConaughey together because I've got an overlap going on. Same difference, I would not want to run a SIM and an indol because you'll notice that SIM does test an indol. And so keep in mind that as you pick your test, you want to make sure that you're not overlapping or wasting test agents. And if you need to review over the results of the various tests and what they test for, again, check out my other 2 videos where I spend a great deal of time explaining each of these test results in addition to the ones on the next slide. And so here are just a few more that I run, and you can see that IMVIC, again, it does the indol again, so you would never want to run this and a SIM or an indol because then you're running 3 of the same test. So hopefully this has helped you with an approach to identifying unknown bacteria, and good luck.